please sit down. Dear guests, my name is Bert Klein-Gebbink and I am the Vice Dean of Education of the Faculty of Science. It is an honor and pleasure to welcome you to this academic ceremony, and I am very happy that on this rather special day we have something to celebrate as well. This afternoon, Professor Villanueva will deliver her inaugural address entitled Ocean's Hidden Heroes, Adaptation and Evolution of Marine Microorganisms. As from November 1st, 2023, Professor Villanueva has been appointed as Professor of Marine Microbiology at the Faculty of Science. Professor Villanueva studied biology at the University of Barcelona, where she did also her PhD. After postdoc positions at the University of Massachusetts and Harvard University, she took a position in 2009 at the Royal Netherlands Institute for Sea Research, known in Dutch as het NIOS. In 2023, she was appointed head of the Department of Marine Biology, Microbiology and Biogeochemistry at this institute. Professor Villanueva works at the intersection of organic geochemistry, microbial evolution, microbial ecology, and molecular microbiology. A lot of words. She's interested in the role of microbes in biogeochemical cycles, their interactions, and the adaptation of microbes to environmental changes. By studying these processes, her work provides new insights into reconstructing past climatic conditions and to predicting the cause of future climate changes. Given her expertise, Professor Villanueva also teaches marine microbiology within biology and geosciences, both at bachelor's and master's level. She is also involved in modernizing the marine biology courses within biology and geosciences. After Professor Villanueva will have finished her inaugural address, you are invited to take the opportunity to congratulate her. Upon leaving this auditorium, please allow the procession and those guests seated on the first rows to depart this room first. Professor Villanueva, please proceed with your address.
Meneer de Rector Magnificus, dear colleagues, friends, family. In the vast, mysterious expanse of our ocean, unseen heroes work tirelessly every second of the day. Meet the marine microbes, tiny but mighty superheroes that dramatically shape our planet. While they might not wear capes or have flashy gadgets, their powers are nothing short of extraordinary. From the depths of the dark ocean trenches to coral reefs, marine microbes ensure that lives persist. They are the ultimate team players, collaborating with ecosystems to break down waste, produce oxygen, and recycle nutrients. Truly, the cleanup crew of the ocean. Like the best sidekicks, they nurture other superheroes, helping them grow strong and thrive. They teach us the importance of connection and collaboration in achieving greatness. It is a great honor to be here today as we embark on a journey through the captivating world of marine microbes. As I stand before you, I want to take a moment to reflect on what sparked my passion for this field. I was 16 years old in high school when I heard for the first time about marine communities, marine microbes collaborating with each other, forming communities in the early Earth. This is what we call stromatolites, which still to this day can be seen in some coast of Australia. These ancient structures are some of the early evidences of life on Earth and played a crucial role in the story of our planet's evolution. These rock-like formations made by layers of microorganisms that date back over 3.5 billion years ago. In a time when the atmosphere had very little oxygen, some green microorganisms, named cyanobacteria, produce oxygen through photosynthesis, transforming the planet by filling the atmosphere with oxygen we all breathe today. As the stromatolites grew, they provided habitat and protected other microorganisms underneath that they could not breathe oxygen, helping them to pave the way to complex life forms, and also showcasing the power of microbial teamwork. This newly found interest in the ecology of microorganisms led me actually to study biology at the University of Barcelona, where I took all the classes in microbiology that I could find. Then I started to collaborate with the microbial ecology group in that same faculty, mostly because they were working with microbial mats, which are the model representatives of stromatolites. A microbial mat is a colorful living carpet made of microorganisms that grow together in layers. The upper layer is green because it's formed by cyanobacteria, microorganisms that photosynthesize as plants. But as you go deep into the sediment, oxygen cannot penetrate, and other microorganisms also doing photosynthesis, but not generating oxygen, as you can see there in red, the purple sulfur bacteria thrive. The red layer is followed underneath by a black layer full of sulfur-reducing bacteria. They produce sulfite, which is the same compound responsible for the rotten egg smell. All in all, microbial mats are absolutely beautiful, layer cakes full of life, where cooperation and competition between microorganisms take place. I remember with excitement the first time that I saw the marine microbial mats at the estuarine of the Ebro River Delta in Catalonia, and I saw and I felt in my hand the bubbles of oxygen coming out of them. They were simply alive, truly the tissue of Gaia. And because of this, I cannot let you go today without telling you about the Gaia hypothesis. Gaia, in the Greek mythology, is the personification of Earth. Gaia is the ancestral mother of all life. The Gaia hypothesis was proposed by James Lovelock in 1972, who suggested that Earth operates as a single, self-regulating system where living things and their environment work together to create conditions suitable for life. This idea highlights how interconnected all living organisms are within Earth's processes. But actually, long before him, Bas Becking, Dutch botanist and microbiologist trained right here in the botanical laboratory in Utrecht University, introduced the concept of Gaia. The Gaia hypothesis as a way to explain the mutual interactions between life and Earth. And he did this actually in his inaugural lecture in 1931 in Leiden, titled Gaia of Leven en Aarde. The interactions occurring in microbial ecosystems, like in stromatolites and in microbial mats, and their role in shaping environments, support the connection between our planet and all living things. Microorganisms, especially marine ones, play also a vital role in the Gaia hypothesis. Despite being tiny, they have a huge impact in the environment by helping recycle nutrients, decompose organic matter, and regulating gases in the atmosphere. For example, they produce oxygen and they help control greenhouse gases. For those who don't know her, this is Lynn Margulis, together with James Lovelock. Lynn Margulis also greatly contributed to the Gaia hypothesis, as she emphasized the importance of symbiosis, how different organisms, especially microorganisms, work together in harmony. Together, Margulis and Lovelock highlighted that every organism, no matter how small, plays a crucial role in maintaining the balance of our planet. Lynn was a force of nature, and my first and ultimate scientific hero. I had the honor to get to know her well during her multiple visits to my PhD supervisor, Ricardo Guerrero, in Barcelona. But sorry, actually I'm getting a bit ahead of myself, because I forgot to mention that I went on and I started my PhD in ecophysiology of microbial mats in the University of Barcelona. From that point on, I was forever hooked up to marine microbial ecology. All of those very early scientific interactions inspired me in more ways than I could think of. The love for microorganisms, for the past, for microbial ecology and the realization that I could be a scientist working at the interface of biology and geology. While microbial mats are fascinating layers of life that demonstrate the power of microorganisms in shaping environments, they are just one example of the incredible diversity and essential functions of marine microbes. These tiny yet mighty microbial heroes play critical roles in the oceans, influencing everything from nutrient cycling to supporting entire marine ecosystems. And as we dive into this fascinating world of marine microbiology, let's take a moment to appreciate just how huge and mysterious our oceans are. While many people picture dolphins and colorful fishes when they think of marine life, today we are going to explore the hidden wonders of the microscopic superheroes that make the ocean their home. Microorganisms are found almost everywhere. In the marine environment, we can find them in coral reefs, estuaries, mangroves, hydrothermal vents, polar ice, deep-sea trenches, on and under rocks, on gills, fins, claws, and even plastics. Those microorganisms living in the marine environment is what we call the marine or ocean microbiome. The ocean microbiome is composed of microscopic organisms, most of which are invisible to the naked eye and largely unknown, even though that they account for more than 2/3 of the ocean's biomass. The ocean microbiome is estimated at 4 gigatons of carbon. That is actually 4 times the overall biomass of insects on Earth. In just one drop of seawater, there can be around 1 million to 1 billion of microorganisms. These tiny organisms, including bacteria, archaea, and phytoplankton, play vital roles in maintaining the ocean's health, supporting nutrient cycling and forming the foundation of marine food webs. You might think that this is a really nice picture of the universe, full of stars, but this is actually a picture of a drop of seawater where marine microbes have been staying. To give you an idea of their importance, just mention that it is estimated that the ocean contains 10 to the power of 29, that is 1 followed by 29 zeros, of bacteria and archaea, and 10 to the power of 30 viruses. That number is so much higher than the number of stars in the universe. But who are they? Marine microorganisms include microalgae, bacteria, archaea, protozoa, fungi. They are incredibly small, often too tiny to see without a microscope. See here this bar at the bottom, for scale, represents 10 micrometers, which is taking 1 millimeter in red there, divided into 1,000 and taking 10. And to help you understand that size, here there are some comparison. You could actually fit hundreds to thousands of marine bacteria or archaea side by side across one of your hairs. Phytoplankton, these plant-like microorganisms that perform photosynthesis, can be a bit larger, but smaller than a grain of sand. And marine viruses, these are among the smallest. To put it into the perspective, a typical virus is about 10 to 100 times smaller than most bacteria. And marine viruses are often smaller than other viruses we know very well, like the COVID-19 virus. These marine microorganisms not only are important in terms of numbers, but they also perform essential functions. They are truly the superheroes of the ocean. Marine microbes are masters at recycling nutrients. They break down organic matter, returning essential nutrients to the water and supporting the growth of larger organisms. They are also oxygen producers. Phytoplankton, the plant-like microbes of the ocean, perform photosynthesis, producing approximately 50% of all the Earth's oxygen. Every breath we take is partially thanks to these microscopic heroes. Marine microbes are also climate regulators. By sequestering carbon and influencing greenhouse gas levels, marine microbes play a crucial role in regulating climate. In fact, marine microbes produce as much oxygen and absorb more carbon dioxide than all of the world's forests. Not only that, but marine microorganisms can also remove carbon dioxide and nitrogen gas out of the atmosphere, incorporating the marine life and further transforming these compounds to fertilize the ocean. They are also the foundation of the marine food webs because they serve as primary food source for other small marine creatures. They support a wide variety of marine life, from whales to fishes. Marine microbes are also disease fighters, as they help protect larger organisms by fighting against harmful pathogens. And they are even producers of beneficial substances. Among these microbial superheroes, we find those producing antibiotics or compounds with the potential to battle cancer, providing healthy fats, vitamins, eco-friendly plastic, and cleaning products we use in our daily life. But marine microorganisms are constantly faced with various challenges, including changes in temperature, salt levels, and nutrient availability. For example, in the deeper parts of the ocean, there's often less light, and the temperature can be very cold or extremely hot near hydrothermal vents. They also need to cope with high hydrostatic pressure. Hydrostatic pressure is the pressure exerted by the weight of water above an object. This has a significant effect on marine microorganisms, especially those living in deep ocean. As you go deeper in the ocean, the pressure increases dramatically. For example, the deepest point on the ocean, known as the Mariana Trench, reaches about 11,000 meters. To put that in perspective, the ocean is as deep as Mount Everest plus 5 Empire State Buildings stacked on top of each other. Marine microorganisms have adapted to these extreme conditions. Some have special membranes that can withstand high pressure, while others have unique enzymes that function well in these environments. To survive in these extreme conditions, marine microorganisms can adapt in several ways. Microbes can change their DNA to develop new tricks that help them survive in challenging environments, just as a person might learn new skills. Microbes can also turn specific genes on and off to adapt to varying conditions, much in the same way we adjust our behavior based on a social situation, such as being more outgoing in a party or more reserved in a meeting. Microorganisms produce different proteins to help them adapt to their surroundings, just the same as people wear different outfits according to the weather changes, just like wearing a raincoat in a rainy day. Microbes also adjust their nutrients based on what is accessible, the same way that we adjust our diet to achieve health goals. Microbes can change their outside membranes to cope with shifts in temperature and salinity, like how we shiver when we are cold or sweat when we are hot. They use signaling molecules to share information and adapt together as a community, the same way we people communicate and collaborate in group projects. They might even produce special compounds to protect themselves from environmental stresses, same way that we exercise or practice mindfulness if we want to manage stress. These adaptations allow microorganisms to thrive where most other life forms cannot survive. Since I started my journey as microbial ecologist, one of the questions that kept coming back to my mind is how microorganisms adapt and how to know if they are affected by everything that surrounds them. I found my answer in microbial lipids, which can sense and change very quickly when conditions change, much in the same way when we get goosebumps when we are cold. But how did microbial lipids come to my life? I was 23 years old, and I had barely started my PhD in the University of Barcelona, working on microbial mats, when I jumped into a plane, first time going to the US, and after 4 different planes of decreasing size, I made my way to Knoxville, Tennessee. At the airport, I was picked by a small lady, Sandy White, the wife of David C. White, who took me out for dinner and gave me the keys of one of their cars. David C. White, DC, was by then in his early 70s, a medical doctor turned into a microbial ecologist, working at the Center for Biomarker Analysis at the University of Tennessee at Knoxville. DC was in love with science, bow ties, and boot turning, one of the most interesting people I have ever met. He was, above all, passionate about lipids. To prove that was the license plate of his Toyota saying PLFA. DC made substantial contributions to microbial ecology. He developed techniques for extracting and analyzing microbial lipids, which serve as biomarkers for identifying and quantifying different microorganisms in environmental samples. There I was, with a suitcase full of little vials with microbial mats to be analyzed for polar lipids, polyhydroxyalkanoids, and more. I spent countless hours extracting, separating, integrating peaks. I wanted to know if microorganisms in estuarine microbial mats felt stress or if their growth was unbalanced during the day-night cycle. DC White believed that research is advanced by using new techniques to study all problems. DC was an amazing mentor who transmitted to me the love for science and lipid biomarkers. I ended coming back to Knoxville up to 3 times. Sandy and DC host me in their house as if I was part of their family. We had breakfast on a kitchen table full of hard copies of several scientific journals, and we had fancy dinners on the weekends where I discovered that I could speak better English when I had had a glass of wine. DC believed that no single technique is enough, highlighting the necessity for new methods to address complex ecological questions. His group started combining lipid and DNA analysis in the same sample, which I also use for my microbial mats. I gotta tell you, I spent way too much time cutting bands of DGG bands like this gel, like this, but it felt great to work with DNA for the first time. In my time in Knoxville, I also met Ronald Geller, a postdoc there by that time. I later visit him when he moved to the UFZ Center for Environmental Research in Leipzig, Germany. Thanks to him, I also dive in the world of respiratory quinones by using LC-MS, again in samples of my beloved marine microbial mats. One of the things I feel passionate about is using different methods. As DC said, no single technique is enough. Microbial lipids are amazing to determine microbial adaptation, stress, and even identify microbial groups, but they are simply not enough when it comes down to knowing microbial communities well. Edward O. Wilson, an entomologist at Harvard, in his book The Naturalist, once said, "If I could do it all over again and relive my vision in the 21st century, I could become a microbial ecologist. 10 billion bacteria live in a gram of ordinary soil, a mere pinch held in between thumb and forefinger. They represent thousands of species, almost none of which are known to science. Into that world, I would go with the aid of modern microscopy and molecular analysis." Molecular methods based on DNA and RNA, but also culturing experiments to determine the physiology of microorganisms are certainly a great option to bridge to the areas that lipid cannot reach. Serendipitously, this is what I end up doing during my postdocs. After I finished my PhD in the University of Barcelona, I moved to the US, to the University of Massachusetts in Amherst, where I did my first postdoc in the laboratory of Derek Loughley, working in anaerobic microbes, those ones that do not breathe oxygen. I did learn a whole lot about anaerobic microbiology, how to handle 20-liter gas cylinders without dropping them, and how to work with RNA. Then I did another postdoc at Harvard University at the Center for Systems Biology, where I focused on projects about adaptive landscapes of proteins with experimental evolutionary approaches. Here you can see where my love for connectors and tubing came from. During my time at Harvard, surrounded by physicists running models all day long, I also came to the realization that what I love the most is an all-good experiment. My time to the US was coming to an end, and I was eager to return to Europe to be closer to my family in Barcelona. When I was scrolling through job offers, I encountered this one, and I knew somehow they were looking for me. How could it be otherwise that there was another crazy person bridging between lipids and molecular approaches? I went all for it. I was so thrilled when I got an interview, only to realize when I was making my travel arrangements that the Netherlands Institute for Sea Research at NIOS was on an island. I didn't know that the Netherlands had islands. My apologies. A city girl, 30 years old, a lock of hair colored purple, ended up in a wooden island full of sheep. A new scientific world opened in front of my eyes. I started my journey at NIOS as a tenure tracker in the topic geomicrobiology, so everything between microbiology, geology, oceanography, and chemistry, always marine, of course, working in an organic geochemistry department. Organic geochemistry studies organic compounds, for example, lipids, in geological context, and how these compounds influence and interact with Earth systems over time. Here, a funny anecdote to the organic geochemist in the house. Claude Sobel, a microbiologist from Scripps, who is considered to be the founding father of marine microbiology, was also fundamental for the field of geomicrobiology and petroleum microbiology. He highlighted the role of bacteria as geological agents in the early 40s, which made a great impression on geoscientists who were convinced they could explain everything in pure physicochemical terms. At the time when the industry was treating the supply of crude oil as an almost inexhaustible reserve, Claude Sobel's thoughts were turning into improving oil recovery by using bacterial products. He also recognized the problem of marine oil spill pollution and the potential of marine microorganisms to help in those situations. This anecdote shows how important it is that marine microbiologists and organic geochemists talk to each other. But let's go back to the microbial lipids. They preserve very well in the sediments, which means that they can be used as fossils of the microorganisms that used to make them. Microorganisms can adapt to the changes in their surrounding environment by changing their lipids, their skin. If lipids change, they sink down the water column, they will deposit in the sediment. They will bury under layers and layers of sediment and preserve as fossils, much in the same way that dinosaurs' bones. Microbial lipids record the conditions at which the microorganisms that carry them lived. For example, the temperature at which they grew. These characteristics make some microbial lipids from marine microorganisms to be excellent biomarkers for paleoclimatology, the study of the climate of the past. Back to circa 2009, when we were a smaller department, Organic Biogeochemistry, BGC, with Jaap Sineke-Damste as department head. I had to get used to very quickly to the brutal Dutch honesty and also to the rural surroundings. Remember that, after all, I'm a city girl. The department was heavily populated by geologists back then, which was a steep learning curve for me. Somewhere between the Younger Dryas and the Hendrix event, the geology seed was planted in me. Maybe it was already there since my time for my love for stromatolites. For the first time in my academic life, I felt my people had my back. The original core group was always there for advice, venting, support, and friendship. If I would need to go through war, I would choose you as my team every single time. The BSc department hosted me as one of them, despite being that DNA lady. I was supposed to bring microbiology and molecular methods to BGC. That is why I spent most of my time in the molecular lab where I felt at home. There, we have experience going from DGE to 454 pyrosequencing, Illumina, metagenomics, to Cascabel, and to an entire bioinformatics team in a blink of an eye. Well, 15 years. My group focuses on the genomic capacity to produce lipids, lipid biosynthetic pathways, namely interrogating microbes for their skills in producing special lipid biomarkers. In a world of marine microorganisms that are difficult to culture, looking at their genes is the best option. There, the GDGT era began. GDGT lipids are specific membrane-spanning lipids found in archaea, of which the marine environment is full of. As they are also used intensively to determine temperatures in the past, the role of my group was to determine who makes them, why, when, and how, by using DNA methods, by targeting the genes involved in the biosynthesis of those microbial lipids. This era began with my first PhD student, Yvonne, and followed by Mark and Martina, who did an excellent job focusing on the membrane lipids of the marine thaum archaeota in different marine settings. Something that I have learned over the years by being at NEOS is that seagoing exploration is important. Especially when it comes to marine microorganisms, we need to continuously improve how we access and study these tiny creatures. Global research expeditions have played a crucial role in this journey, allowing us scientists to collect samples of marine microorganisms from various locations around the globe. Seagoing research is not only fun and an interesting exercise of teamwork, but also we should see it as an immense privilege. I have been honored to be part of 3 expeditions so far during my time at NEOS, and that is only possible with the immense support of the NMF department at NEOS and the crew and scientists on board of the RV Pelagia. In the coming years, further advancements in marine microbiology will also depend on improvements in collecting data and samples of marine microorganisms that are yet non-accessible or we have not there found them. In general, we still lack sampling and incubation instrumentation that will ensure conditions as the one those marine microbes normally live on. Another scientific revolution that has significantly impacted the field of marine microbiology in the last 10 years is the application of molecular methods. Studying marine microorganisms using molecular methods is like being a detective trying to solve a mystery by carefully examining clues. By reading, or what we call sequencing the DNA of a marine microorganism, we can determine who it is and what activities might be performing. Now, we can even do what we call metagenomics, which implies sequencing the DNA of different microorganisms, including an environmental sample. Microorganisms are everywhere in the marine environment. There are some that are still quite unknown to us, mostly because they are difficult to reach or to work with. In deep sea sediments and in marine areas where oxygen does not reach, we find some marine microorganisms which have been traditionally quite neglected. Breathless or anaerobic marine microbes that do not breathe oxygen like us, but something else. These marine areas are called dead zones because most marine life, like whales and fishes, die. Well, for anaerobic microbes, this is the perfect playground. Lack of oxygen or marine deoxygenation is mostly caused by warming due to climate change. Pollution and overfishing, all of this caused by us, humans. Climate change is caused by human activities that release greenhouse gases into the atmosphere, primarily caused by burning fossil fuels, deforestation, farming, pollution, and landfills. This is causing rising temperatures, extreme weather, melting ice, higher sea levels, which threaten ecosystems, food supplies, and access to clean water. In the last weeks, we have witnessed intense rainfall and flooding in Spain that has come with destruction and human loss. Due to the very evident consequences in Spain, people are now believing that climate change is real, which is not something so evident for others living in other parts of the world. Are we still in time to do something about it? Or that a climate crisis is serious is something that you only realize when it's too late? When it comes down to marine microbes, What can we do as scientists? First, to know how they are affected by climate change, because that affects the entire marine ecosystem. And also to change the focus to anaerobic marine microbes that live in oxygen-depleted areas of the ocean, because those, they have the potential to further contribute to climate change by producing greenhouse gases. With this in mind, a great opportunity came to our door in 2013 when we got an NWO gravity grant to work together with the best microbiologists in the Netherlands as part of the Zoingen Institute of Anaerobic Microbiology, SIAM. The role of NEOS in SIAM was to study anaerobic marine microorganisms with emphasis on those involved in the cycling of carbon. During my postdoc in the University of Massachusetts, I had gained a lot of experience in growing anaerobic bacteria, so setting up a lab at NEOS for anaerobic microbes was a very exciting experience. Then, we quickly started hiring very talented PhDs and postdocs, working on microbial lipids and also on marine microbial physiology. We started sampling Lake Grevelingen in the Netherlands, but even went to the remote Black Sea in several occasions. Experiments were conducted on board of the RV Pelagia, and very precious samples brought back to continue working with anaerobic microbes from the deep waters and sediments of the Black Sea. Several theses have focused on anaerobic marine microbes involved in the cycling of organic matter by Sarah, methylated and sulfur compounds by Peter and Dan, and in the methane cycle by Sigrid, some in collaboration with amazing SIAM partners of Wageningen University. SIAM has not only been an amazing source of funding for 10 years, but also a once-in-a-lifetime opportunity to advance the field of anaerobic marine microbiology. It also helped in recruiting and training the next generation of marine microbiologists, not only PhDs, some of them here today, but also international students, thanks to the EMBO Summer School on Breathless Microbes. Another focus of the work of my group in the past years also involves how microbial lipids have evolved and why we should care. When looking at microbes and their membrane lipids, we can search if they have the genomic capacity to make them. Same way that if I would go to your house and check if you have Spanish books in an attempt to decipher if you are able to speak Spanish with me, I can also interrogate microbes to their ability to make a lipid. When we look at the genes of microorganisms, we open a book that tells us a story. How do they make these lipids? Since when? What happened during the course of evolution? Membrane lipids of the cells of bacteria and archaea are structurally very different, and also probably a reflection of the adaptation mechanisms that they had to acquire over time. That leaves us the question on which kind of membrane lipids, which kind of skin, the last universal common ancestor look ahead, and how this differentiation or lipid divide took place. In this kind of research, we have combined lab experiments with omic approaches, and we have even dived to the bottom of the Black Sea looking for answers. The ocean is not only the playground of marine microbes, but also hides many secrets of how life appeared and evolved on planet Earth. All in all, after introducing to you the amazing microscopic heroes living in the ocean, their adaptations, their hopes and dreams, their lipids, I hope I have convinced you by now that having a special chair in marine microbiology at the Faculty of Science in Utrecht University is really important. The focus is on marine microorganisms from the past to the present to the future, understanding their diversity and their roles in natural processes, which is crucial in tackling climate change effects on marine life. This position aligns with Utrecht University's commitment to sustainable development goals related to ocean protection and climate action. I'm deeply honored by the opportunity to serve as a special chair of marine microbiology in Utrecht University. I cannot wait for the new collaborations and opportunities that will strengthen the collaborations between the faculties of geosciences and science and the collaboration with NEOS. And of course, contributing to educating the next generation about the importance of the oceans. The future of marine microbiology looks bright. However, many challenges still remain. Public engagement in marine microbiology is crucial because it raises awareness of the vital roles that microorganisms play in the ocean's health. As scientists, we have the responsibility to reach out to non-scientists, to all ages and levels, and to learn how to bring the message across, not just from a podium, but also in schools, in collaboration with artists, journalists, and in community events. Looking ahead in the future, another major challenge is how to support marine microbes in their quest to support biogeochemical cycles in the ocean. While microorganisms are essential for maintaining Earth's balance, their ability to cope with rapid climate change is limited and threatened by us humans. To protect our planet, we must work together to reduce our environmental impact and support marine microbes and their ecosystems to ensure a healthy Earth for the future. It's time to take action and safeguard the environment that sustains us all. Here we go. Acknowledgements. I'm beyond words to explain how much it means to me to be here today giving an inaugural lecture in this room in Utrecht University in the Netherlands, a country that has fostered the best microbiologists in the world and has given me excellent colleagues, friends, and my own family. Now it's time to say thank you. I feel like someone accepting an Oscar award and trying to thank everybody as quickly as possible without forgetting anyone. So here we go. First of all, I'm very grateful to the Executive Board of Utrecht University with the President, Professor Anton Pijpers, and the Rector Magnificus, Professor Henk Kummeling, as well as the members of the Board of the Faculty of Science with the Dean, Professor Isabel Arens, for establishing the Chair of Marine Microbiology in the Faculty of Science. I would also like to take this opportunity to thank Professor Han Woesten for nominating me for this chair, and also to many new colleagues at the Department of Biology at the Faculty of Science for their support. I would also like to thank the previous and current directors of NEOS, Professor Carlo Hype, Professor Hen Brinkhaus, and Professor Han Dolman, respectively, for their support and advice. I thank all the colleagues of NEOS in general for their help in the last 15 years since I arrived at NEOS. A right is not worth taking unless you do it with the best people. A big shout out to the PIs of MMB for their endless support. And also to the MMB assistants for the trust they put in me every day and for their support as well. I would not be here today without these two. When things get tough, I always channel the inner Jap in me. When I find myself at a crossroad, I always wonder what Stefan would do. Thanks. I would like to thank the members of my group, Karen and Paz, some present here today and some gone. You are an endless source of inspiration and my hands and my brain pretty much all the time. I would like to thank all my academic colleagues in the Netherlands and abroad. And I would also like to thank the SIAM community with a big thank you to Mike for steering the boat, and especially to the SIAM ladies for being amazing colleagues and friends. And of course, a special shout out to the members of the KNVM board, iedereen heel erg bedankt voor de gezelligheid. I would like to thank all my friends here in the audience and online for being always in touch, sometimes even from the distance, through never-ending WhatsApp podcast. Especialmente a mi mejor amiga, simplemente no hay palabras. Ik ben ook zo dankbaar voor mijn Nederlandse familie. Bedankt voor jullie steun en liefde. Y a mi familia en Barcelona, gracias por estar siempre ahí, por todo vuestro amor. Thank you also, Wapo, for all your love, for supporting me in everything I do. You're my everything, and the best partner to go through all the chaos. I cannot believe we went through here to here. I cannot wait to see what is yet to come. And Noah, Alex, y Zoe, los quiero. Thank you to all of you here and also online. Gracias, gracias, thank you, bedankt. And to close up and compose myself, next time you gaze at the ocean, remember that beneath the surface are endless stories of heroic marine microorganisms, diligently working behind the scenes. They remind us that we can be impactful regardless of your size, and that every contribution, no matter how small, plays a vital role in the health of our planet. Ik heb gezegd.